Thanks very much to the organisers for inviting me. It's a great pleasure to be here, and thank you all very much for coming along. We really have a lot of super exciting eye research going on at the Royal Adelaide, and I'm delighted to be able to share just some of that with you this evening. First, let me take you on a brief journey through the eye. Vision is our most precious sense, I think you could say. And the eye is said to be a window to the soul. Well, it's certainly a window to many of the ailments of the brain and body. And it is, of course, our aspect to all the beauty that we see in the world around us. The workings of the eye are truly an absolute marvel of nature. The optical elements at the front of the eye, the cornea and the lens, work together to focus light onto the light-sensitive film at the back of the eye, the retina. And there the light is converted to electrical signals which are transmitted along the optic nerve to the rest of the brain. Well, why are there visually impaired people then? Well, either because of limited resources or increasingly untreatable disease for age-related conditions like macular degeneration or glaucoma and genetic conditions like retinitis pigmentosa. But there are emerging treatments, new microsurgical techniques at the absolute microscopic level, new imaging techniques, new laser technology to treat glaucoma and diseases of the retina, and to treat disease at the subcellular level. New proton therapy coming to South Australia to treat eye cancers, new genetic discoveries, new gene therapies, and artificial intelligence and machine learning that lends itself so well to eye disease, and new drug discoveries, and new bioenergetic methods at the subcellular level. So it's certainly a really exciting time to be involved in ophthalmology and eye research. Let me just take you through again briefly the workings of the eye to give you a little bit of a background to what I'm leading up to here. So the light is focused by components of the front of the eye onto the retina. It's a beautiful laminated structure at the back of the eye, and then information travels along the optic nerve. That panel on the right is the optic nerve as we see it clinically when we're looking into someone's eye with an ophthalmoscope. Now, light strikes those cells there that just lit up then. They're called photoreceptors. They respond to light. They send information to a middle layer of cells in the retina that then sends information to these guys here. The blue cells are retinal ganglion cells. It's those cells that die off in glaucoma, and their nerve fibres comprise the optic nerve which runs along to the brain. So the visual pathway is an absolutely beautiful structure that extends all the way across the brain from front to back. The retina itself is actually part of the brain, extending all the way across to the far back of the brain. A little bit more detail on how these photoreceptor cells work along the right there. These guys are called cone photoreceptors. They respond to blue, red and green wavelengths of light and give us our colour vision. These guys are tightly packed at the centre of the retina at the back of the eye in a region called the macula. It's that tight packing that gives us our fine vision to see faces and for reading. They contain a convoluted membrane within them that contains many different little proteins that respond to light. Each photon of light changes the shape of these proteins. When they stop working, we stop seeing. They have a counterpart in the retina called a rod, which gives us our night vision. The rods can respond to up to a single photon of light. In a disease known as retinitis pigmentosa, it's those rods that become damaged through genetic disease. They stop working, and so loss of night vision is one of the first symptoms. The retina requires enormous amounts of energy to keep vision functioning. This is why we're so interested in energy metabolism. So it's very prone to any problems of energy metabolism that might occur. I've shown a picture of the sun there because almost all life on Earth ultimately derives its energy from the sun. And in fact, all life on Earth uses this molecule here, ATP, as its energy currency. So that's a super important molecule. Just a little bit of chemistry here is that all cells make ATP molecules by two methods only. This is the first method that doesn't necessarily require oxygen, called glycolysis. It's exactly the same as fermentation. Sugar, glucose, is converted to a molecule called pyruvate. Each glucose molecule makes 2 ATP molecules in this case. Pyruvate is then converted to lactate, and when oxygen is scarce, the lactic acid builds up. So that's exactly what happens to athletes when they've been running too far and running out of oxygen. A special molecule, helper molecule called LDHA, is required for the conversion, and lactate in some cells can be converted back to pyruvate with an LDHB molecule. To keep the whole thing flowing, this substance here, NAD, is critical, which is a derivative of vitamin B3. More of that later. Pyruvate then enters into, these little critters are mitochondria inside all our cells, or almost all our cells. In the presence of oxygen, they convert glucose to 32 ATP molecules. So they're way more efficient, but they require oxygen. So I'm involved in research in the laboratory at the Adelaide University, the ophthalmic research laboratories, and also the eye department at the Royal Adelaide. And we engage in so-called translational eye research. It's what it's all about. So we aim to bring new discoveries in the lab as rapidly as possible to the clinic. And I'll be mentioning a couple of those in a moment. We have national and international collaborators, and I think it's fair to say that we're world leaders in this area. I'm a glaucoma subspecialist. So glaucoma, it's a disease I'm particularly interested in and finding new treatments for. It's a group of conditions where the pressure in the eye damages the nerve of sight. The treatment revolves around lowering the pressure in the eye, and in most people that is successful if it's diagnosed early enough and treated properly. However, in some people, even with our very best efforts, vision continues to be lost. So we'd desperately like to discover new treatments that we can add to our current treatment program. Over time, the vision eventually caves in, and at the very end stage of glaucoma, all vision is lost to total blackness. Retinitis pigmentosa, as I mentioned, is a condition where the rods degenerate and has a similar effect on the vision over time, starting with loss of night vision and then tunnel vision and eventually loss of all vision. The cones, although not affected by the abnormal genes, eventually die off in most patients with retinitis pigmentosa, and all central vision is lost. So there's good evidence that both glaucoma and retinitis pigmentosa are at least in part caused by an abnormal energy metabolism or insufficient energy supply. So we've been studying this for some time in our laboratory. So John Wood is a senior researcher in the labs. I poached him from Oxford University about 15 years ago, and he's still with us. John's a world authority on growing retinal cells in culture. Those are retinal ganglion cells that John has grown in culture. And it's important to bear in mind that the retina has the greatest energy demands of any tissue in the body. That's why energy metabolism is so important. Now, the Warburg effect is an extremely curious thing that I just wanted to mention, and this was discovered by a guy called Otto Warburg in the late 20s. He won the Nobel Prize for this. What he noted was that cancer cells tend to make energy via the glycolysis mechanism, turning glucose into lactate, despite the presence of oxygen. This was a very strange thing, why they would seemingly sacrifice the ability to make lots of ATP and instead make a tiny amount of ATP each time from lactate. It's sometimes said to be a hallmark of cancer. That's not true, because the retina has exactly the same phenomenon. Photoreceptors also like to turn glucose into lactate, even in the presence of oxygen. It's a very strange phenomenon that we don't understand, but it's certainly curious, and the link between the photoreceptor metabolism and cancer is quite intriguing, and I think deserves a lot more study. We've published on this in the past and have just sort of cautioned some of the cancer researchers that they will need to be careful targeting cancer metabolism as a potential treatment, because anything that potentially affects cancer metabolism could also affect the photoreceptors of the retina, inadvertently, obviously. Just moving on then to think about retinal nerve health over time. I'm thinking here about the optic nerve. In a chronic disease like glaucoma, it slowly dies off. The nerve fibres slowly die off over time. Most of our techniques in detecting this, particularly the current imaging techniques, although they're excellent, only really detects fibres that have degenerated. So once they're lost, we can see that. What we would really like to be able to do is detect cells that are sick way before they're degenerated, to institute treatment at a really early stage. Now, to that end, we asked the question a few years ago, well, can we use light to try and capture information about retinal metabolism? The eye lends itself beautifully to light and laser technology. It's just ideal for that. And of course, there's so much of that happening now. So we've been utilising the property of fluorescence, which many cells have, and in fact there are many molecules in the retina which fluoresce on their own accord automatically. And we've been collaborating with Eva Goldis, who's Professor of Physics at the University of New South Wales, to develop a new retinal camera that captures energy information about retinal fluorescence. So that's ongoing work, and that's really bringing space science technology to the eye. We're also interested then in the concept of delivering energy to the retina, to the hungry retina. This was an experiment, those top panels that we did a number of years ago. The sort of starry sky red stars there are retinal ganglion cells from a rat retina. Now, on the control panel is a nice healthy one. The middle rat, had glaucoma, and his retinal ganglion cells are dying off. That's why he's got less red stars there. His friend had exactly the same glaucoma, but he's been delivered glucose to his retina at the time of the injury, preserving the retinal ganglion cells. So that was quite a marked discovery in the laboratory, which we took to a trial in patients here at the Royal Adelaide Hospital a few years ago to demonstrate essentially a proof of principle concept that delivery of an energy source to the retina could, in this case, actually temporarily recover vision in patients with glaucoma. During this time, one of our other researchers, Glyn Chidlow, who I also poached from Oxford the same time as John 15 years ago, and he's still here with us. Glyn is, I think, the world's best retinal immunohistochemist. It's a method used in the laboratory to stain particular cells in the retina. And Glyn has demonstrated that the retinal ganglion cell, we were asking the question, well, what do they actually like to eat? What would be their preferred food to eat? And it looks like they can uptake lactate and convert it to pyruvate. So that was an extremely interesting finding. Around that same time, this young fellow published what's now become quite a famous paper in Science. The journal Science is one of the highest ranking scientific journals in the world. And he found that vitamin B3 massively protected mice against glaucoma. And remember that vitamin B3 is required in glycolysis. So this was really interesting. And I met this young fella just before he published it. And we realised we were working on similar things. And we then went on together to look at pyruvate as to whether that could protect cells. John Wood showing that first, that yes, pyruvate did in fact protect cells against an energy deprivation injury. And then one of our master students, Chelsea Geimer, in the lab, then went on to show in animals that pyruvate was quite markedly protective against a glaucomatous injury. Pete, also in his laboratory, was doing exactly the same thing in a different model of glaucoma, finding the same results. So that was really encouraging to have two different animal models of glaucoma, both showing the same thing. So that paper is in preparation as well from that data to send to Science as well, actually. And in fact, researchers in New York have already saw us present some of this at a meeting last year and have already started a clinical trial of vitamin B3 and pyruvate in New York. So they're going to beat us to it, I think, but we would have liked to get there first. Chelsea also demonstrating that the optic nerve was preserved. A colleague and collaborator, Jonathan Croston, was in Melbourne, now in Singapore. We collaborated with and showed that vitamin B3 did in fact temporarily recover electrical activity of the retinal ganglion cells in human patients over a short time period. So that was really exciting initial work. It will take a longer clinical study to show that it really preserves vision over time. This young fella, Mark Hassel, is a scientist and an eye doctor. He's training to be an ophthalmologist. He's currently working with me in the glaucoma division. He's a super smart guy. He was a Rhodes Scholar, did a gene therapy PhD in Oxford. He's now back with us, so it's great to have him at the Royal Adelaide Hospital. He's studying this thing here called neuroglobin. Now, you might have heard of hemoglobin is the molecule in the blood that carries oxygen. There's a molecule in the brain and the retina that also carries oxygen called neuroglobin. And Mark has been doing experiments to try and upregulate neuroglobin in the retina. And he's had some really exciting results around that. The retina needs enormous amounts of oxygen, more per unit mass than the brain. And the lighting up starry sky there is basically Mark demonstrating that he's delivering his gene to the correct spot, to the retinal ganglion cells. And we've already have data indicating that he's getting some protective effect on it in glaucoma models. So that's really exciting. This is ongoing work. The final thing I wanted to show you is a really exciting development there's a thing called photobiomodulation. That is a low energy light to treat various diseases, and it acts on the mitochondria, those cells that make energy. As it turns out, an Adelaide-based laser company made a laser for ophthalmic use that delivered the exact wavelength that we wanted to use. There's only 3 of these lasers, 3 prototype lasers in the world. We've got 2 of them. One's in the clinic at the Royal Adelaide, one's in the lab at Adelaide Uni, and one's in Sydney. Mark Gillies is our collaborator in Sydney, and using this laser, we initially showed that it reduced leakiness to blood vessels, and then in collaboration with Mark, we showed that it reduced thickness of swelling of the retina in diabetes. That was in human disease, a small phase 1 trial, so that's a really exciting result. We later found in the laboratory that it had this same laser protected cones in an animal model of retinitis pigmentosa. We then took it rapidly to a clinical trial, and this young lady is receiving the laser treatment here to the back of her eye. This young lady has very severe retinitis pigmentosa. She only has a small amount of central vision remaining, and it also happens she's totally deaf. She has a condition called Usher's syndrome. So we're desperately trying to preserve her remaining small bit of central vision. I treated her friend today earlier at lunchtime with the same condition. These were the laboratory results. Again, the sort of starry sky pattern is that basically we were rescuing these cells called cones in the animal model, and the effect was quite so massive, we thought we need to get this to the clinic pronto. And so we were given ethics approval by the RAH Ethics Committee. We've just completed the phase 1 trial. It was only a small number of patients, 12 patients. You guys are the first in the world to see this data then. But basically what we're showing there, the sort of upgoing graph like that on the right, is a temporary recovery of vision, of the ability to read the eye chart in these patients. So they were all excited about that. So this is work that we have to continue somehow. So we desperately need funding to extend this to a larger study, to a phase 2 trial with more patients. So I'd like to acknowledge my team there in the lab and at the Royal Adelaide Hospital. And thanks especially to the RAH Research Fund and everybody for organising the event. And thank you all very much for coming. We really appreciate it. We desperately need ongoing support. So I'd like you all to please consider supporting this research. Thank you very much.
I have a few questions from the audience that I'll go through, and then we'll open up the floor a little bit later after the filming. So one of the first questions, though, is do we know what actually causes glaucoma? You touched on the increased pressure in the eye.
Yeah.
Is there a problem of blood supply to the back of the eye that stops the oxygen getting through as a result of the pressure? It's just, I'm making that up.
Yeah.
Is that
No, well, that is thought to be a possibility. So we don't actually know what causes glaucoma. So we know that the pressure is important, and the current treatment all revolves around lowering the pressure in the eye one way or another with drops or laser or surgery. The 2 main theories that have been around for a long time around glaucoma, one is a relatively simple idea that just the pressure, the mechanical pressure in the eye damages the sensitive nerve fibres. The other long-standing theory is that there is in fact impaired blood flow to the optic nerve, and that lowering the pressure in the eye improves the blood flow, reduces the resistance to the blood flow. So in all likelihood, it's probably the case that there's different factors affecting different patients. And at the moment, it's difficult to tease that apart, but that will come.
I'm aware that one of the treatments for glaucoma involves surgery to, make a little hole to help sort of relieve the pressure, and maybe some of the people in the audience may have even had that done. I'm not sure. Why doesn't that work in everyone?
Well, that's a good question. I guess there's two reasons why that might not work in everyone. One might be that the surgery does not lower the pressure sufficiently, let's say. You could make the case that if you could lower the pressure low enough, it would work. But it looks like that there are some people, for unclear reasons, perhaps because the blood supply or actual intrinsic energy metabolism is so dysfunctional that no matter how low you lower the pressure, they still continue to progress. The percentage of patients that happens to is hard to say. probably less than 15% around there, but maybe at least 10% of patients. But of course, they're the ones that end up with progressive disease.
Sure. And I'm aware that yourself and some other very prominent ophthalmologists in Adelaide have done a lot of overseas work to third world countries. Just looking at the sorts of treatments that you're talking about here, perhaps putting aside the laser, they sound relatively inexpensive. Are these things, correct me if I'm wrong, that could be widely applied, these sort of glucose modifying things?
Well, yes, the new treatments that we've got in mind could be widely applied. So our strategy is to develop treatments that we think will have minimal adverse effects, in a way almost repurposing available drugs or energy supplements that can be brought to clinic very quickly and trialled in small phase 1 trials and brought to larger trials quite quickly, yes, and relatively inexpensively.
And in a couple of your descriptions of your experiments there, you talked about temporarily improving the metabolism. Why is it only temporary? Is it just because you need to give it for longer, or is there something else that happens?
It looks like you would need to give it for longer. Would need to be an ongoing treatment for a chronic disease. And in terms of that laser photobiomodulation, it looks like that the effect wears off over time. That the effect that the photons of light have on the basically the atomic level in the mitochondria wears off over time. It's thought that it soups up the ability of the mitochondria to make ATP, and those little critters, the mitochondria, also make a lot of pollutants. They're kind of like an engine. So they make energy, but they make pollutants as they're making energy, and the laser is also thought to mop up the pollutants as well.
And I was also fascinated about the gene therapy approaches, because I've done a little bit of that in the lung myself. And with the approaches that you're using, adeno-associated virus, is there any immunological problems, or is the eye relatively protected from that sort of thing in terms of using these?
Well, that's viruses? A good point. There's not that we know of with that particular virus that we use. The adeno-associated virus has very low immunogenicity, but we are careful of that. So we looked extensively to see if there was any toxicity from that approach, and as far as we can see, it's completely non-toxic.
Yeah, that sounds excellent. Well, a couple of other questions. Are there other retinal diseases that actually have impaired energy metabolism beyond glaucoma?
Yes, so a number of diseases, in fact. So there are obviously, the diseases where the energy metabolism impairment is an obvious factor, and that would be diseases where the blood supply is impaired to the retina. So you could have a condition whereby the arterial supply was impaired, let's say, or more commonly, the drainage supply, the venous supply, can be disrupted in the retina with things like what's called a branch retinal vein occlusion or a central retinal vein occlusion. These are quite common conditions with same sort of risk factors as cardiovascular risk factors, same risk factors as heart attack, causes the veins to block in the eye. So they're definitely accompanied with energy impairment. Other conditions that are probably also accompanied would be things like macular degeneration and also diabetic eye disease.
And looking at the experiments, perhaps I was wrong, I understood that most of the treatments were being topically directly into the eye, but the vitamin B3, is that something that can be taken orally?
Yes, so B3 was orally in quite high doses in the mice. I'm in fact already recommending it to a number of my patients in certain doses. It looks to be very safe. Right. There's no definite human data yet, but because it looks so safe and the results were so promising in the animals, I think if I had certain forms of progressive glaucoma, I think I'd be taking it myself.
Right, okay. And with the retinitis pigmentosa, which is obviously a different condition, I mean, what's the current state of the art there? I mean, you've touched on it a bit there, but maybe just expand on that a little bit.
So it's a whole group of genetic diseases that predominantly affect these cells called the rods in the back of the eye that provide our night vision. The genes in the cones are not affected, but yet in most people the cones eventually die off for reasons that are unclear, possibly thought to be an energy problem. The state-of-the-art treatment, well, for a sort of a handful of the genetic causes of RP, there's now gene therapy treatment. So the FDA, the American approval body, recently approved gene therapy for one particular form of RP. The Bionic Eye program has essentially been directed towards retinitis pigmentosa, so artificial retinas is basically for RP. But really, basically, there's still really not much.
Yeah. So someone actually had raised the bionic eye, and what is the state of art of that? How well can these things work? Can people actually see?
Yeah,
with these
it's
things?
been around for a long time. It is improving over time, and there's been various strategies to this. When people talk about a bionic eye, what they're really talking about is an electronic retina, a portion of the retina is replaced by electronic circuits that respond to light and then send information to the remaining retina, which is hopefully still alive. So those photoreceptors at the back of the retina are degenerated. The electronics try to substitute for that and send information to the other layers in the retina. Now, even with current technology, the amount of vision you attain from that is quite poor. Nothing like reading, let's say, the top letter on the chart. It's a long way from that still.
Okay, so lots more to be done.
Yeah.
Someone's raised a specific question about Bardet-Biedl syndrome, which is associated with retinitis pigmentosa. Perhaps your previous comments sort of cover this as well, but is there anything specifically in that syndrome that's a bit different?
Not specific to that. So Bardet-Biedl syndrome is a condition occurring in childhood usually where there's a variety of disorders, a variety of things not working correctly, including the rod photoreceptors in many people. But the underlying cause and pathology of that's not well understood, so it's not seen as really distinct from the other batch of retinitis pigmentosa, and as far as I know, no specific treatment.
Sure, okay. One of the other questions was how important is philanthropic funding for what you're doing? I know we all struggle with government grants. How much of your work relies on that side of the equation?
Well, I'm glad you asked that one, Paul. Yeah, that grant funding, as you know, John and Andrew alluded to, is becoming increasingly difficult. Very difficult to get funding these days from the government, from the NHMRC or other sources. So we rely heavily then on philanthropic funding. But basically, without that, the research doesn't keep going.
Mm-hmm. And looking at the translation to clinical trials, I mean, on this campus, I mean, you've already shown us some early trials. Do you see that progressing into the future? Is that rapid translation something that we can continue?
Yeah, the thing I'd like to do next would be a larger trial of the retinitis pigmentosa laser.
Yep.
A trial of the pyruvate in glaucoma, possibly combined with the vitamin B3, see if we can beat the New York team to it. There's a variety of other, I showed you really just a portion of stuff we're doing in the lab tonight that we want to take to the clinic. So there's a whole bunch of stuff to do. Yeah.
Excellent, great. Well, we wish you every success in that. And look, that concludes the formal questions that I have.